Hello, everyone. I'm going to talk to you today about aging. In particular, I'm going to talk about the science of aging. And the subjects interested me as soon as I realized, as most of us do, that at some point or another, usually at an average of 75 years of age, we're going to develop some sort of disease that will end up being fatal. And we are the only species that is able to, despite their biological imperative to stay alive, to realize that it's ultimately a futile effort. And although it is a frightening thought in the beginning, I think it's one that brings us character, and for most of us makes us dedicate our lives to something bigger. And when you think about it, when you age, you're going to develop some sort of heart disease, neurodegeneration, or cancer that will ultimately be fatal. And I knew that there were a lot of scientists that are devoting their lives into finding treatments and cures to these diseases. And what to me was the fundamental problem was that even if we find a cure for these diseases, no one escapes aging. And for that matter, would you really want to extend your lifespan, even if it's free of these diseases, having the quality of life that is associated with the deterioration that comes with aging? And so this is ultimately what got me interested in aging and stories like the one from John Calment. And John was born in 1875. Her family owned a business store, a store at which at age 13 she met Vincent van Gogh as he came into her uncle's store. Around the same time, the Wright brothers were making history in aviation, and she lived through some of the most amazing discoveries that were ever done by mankind. She witnessed the introduction of the first Model T automobile. Over the span of the next 4 decades, she lived through 2 world wars, and over the lifespan of over 100 years, she witnessed the evolution of technology that she saw first introduced. John rode her bike until she was 100. She died in 1997 at 122 years of age, having beaten the record for the world's oldest person. And if you're interested in what were a lifespan choice or lifestyle choices that allowed her to reach that age, well, she was said to like chocolate. She ate 2 pounds a week, and she quit smoking when she was 117. And so, John is really an exception of longevity, but to me she's an example of how your genetic makeup can determine how long you live and can determine how healthy you live, your lifespan. And why can't we all be like John? Why is it that some of us have this lifespan of 75 on average, and why is it that people like centenarians can live for 100 years and live so healthy? And for that matter, what interested me is, why do we even age at all? And that was a question that was first introduced by the father of biology, Aristotle, in 350 BC, where he said that he noticed that individuals within the same species have different lifespans, but also that different species shared very different longevities. And so what he was asking was essentially, why is it that dogs have a maximum lifespan of 25 years, similar to that of cats that are 30 years? Why is it that we have this cap at 120, yet organisms like a bowhead whale can live for over 200 years? And we know today that we age because we have different genes that we have evolved over the course of our evolution that have some detrimental effects late in life. And what I want to tell you about today is a key discovery that was done in first identifying the genes that are responsible or that are able to influence the process of aging. And it was a discovery that came from worms. It wasn't any worm. It was a very specific worm called C. elegans. It is famous among geneticists around the world. That's how it looks. It is famous because it is tiny. It has about 1 millimeter in length. It only has about 1,000 cells, and we know the position of every cell. It was one of the first to have its genome fully sequenced, and a lot of genetic manipulation is possible in this organism. And importantly for the study of aging, it lives for about 15 to 20 days. So we could do a lot of experiments quickly in this animal. And when we first started working with it, some researchers found that when conditions were slightly off, when the conditions in the plate weren't good enough, the temperature was slightly off, or there was not enough food, they entered an alternate stage that looks very similar, slightly smaller and thinner, called a dower. Now, a scientist by the name of Michael Klass in 1976 was studying the biology of aging in these worms. And he found that these dowers, while a normal worm lived for about 15 days, these dowers lived for 3 months. And that was an amazing discovery in its simplicity. Not only that, he showed that there were no detrimental effects of aging while these animals were dowers. And what that suggested is that there ought to be a genetic program that is able to influence how long animals live. And so he set out to look for these genes. And the way we do that in the lab is to randomly take a gene here and there from this organism and look for the effects in lifespan. And he found such a gene. He found a gene that when the animals have reduced activity, it extended their lifespan almost to double. And it was recently shown that when you fully take out this gene, the lifespan of these animals is increased by 10 times. That is the difference between a worm that lives for 15 to 25 days to live now 9 months. And it will be the equivalent of us living for around 750 years. And we named this gene appropriately age 1. And the question that we had now was, all right, we can extend the lifespan of these animals. And any scientist at the time will tell you that that's not possible with a single gene mutation. And that's what he showed. Not only that, he showed you can look for them using the same approach. And so we found, taking similar approaches, now over 400 genes that are able to influence lifespan. And the question that we had was, can we do this in other organisms? Could we use the things we learned from this roundworm to use it to mammals or even to us? And when people tried, first, age 1 is actually a molecule that is in the pathway that regulates the response to insulin. And consequently, we and mice as well have it. And so when downregulation of that pathway was tried in mice, a similar lifespan extension was observed. And we now know that centenarians like John Coleman have very particular forms of the genes in this pathway that are likely to be responsible for their longevity. And so this, a wealth of work has been done to show that, before that, there's a fundamental difference between C. elegans and mice that I forgot to tell you about. And that's that when C. elegans develops, their cells divide, and when it reaches adulthood, they stop dividing. And so they can't form tumors. Yet mice, for example, they frequently form tumors, and they die from them in the lab. And so how is it that an intervention that is able to extend the lifespan of a worm is able to extend the lifespan of a mammal with such different pathology? And that was a question that was asked after this, and that would shape the way that we see the biology of aging. Could it be that an intervention that extends lifespan also postpones cancer? And so a wealth of work has been done in this to show that that's exactly the case, is that we now think that aging is the fundamental cause of some of these diseases. And if by altering the rate at which you age, you are able not only to postpone the onset but the progression of disease. And that was part of the work that I did during my PhD. I reasoned that if aging was really at the cause of disease, then we ought to see some of the same molecular mechanisms that we see in disease happen in aging. And in particular, I was interested in neurodegenerative disease. And as you heard before today, in the brains of these patients that have Alzheimer's, Parkinson's, or Huntington's, there's the formation of these plaques that are formed by a process that we call protein aggregation. Protein aggregation is essentially the proteins inside our cell coalescing together to form large, inactive forms that are frequently toxic. And what I have observed using these animals, taking proteins from old and young animals, is there are about 200 proteins in these animals that share similar characteristics with the ones from the Alzheimer's. And so what does that mean to us? It means that in the biology of aging today, we have a grasp on the causal relationship between aging and disease. It means that we now think that aging, which is caused by molecular damage to our cells, leads to the tissue dysfunction that allows for the onset of disease. Lastly, I would like to say that the biology of aging is not about trying to make you live longer. It's about trying to make you live youthful. It's about postponing disease. And that the study of the small roundworm was only the beginning, and we need to do a lot more work into understanding the fundamental mechanisms of aging. And although the transfer of knowledge to technology is really ultimately what we're looking for, there's still a lot of work to be done. And I believe that we're heading to an era in which we have finer and better control of our genetic makeup, and in which the translation from knowledge to technology will become faster. Imagine what we could do with this knowledge. Thank you.